Okay, yeah. So hello, everyone. Thank you for joining us to attend this workshop. So this workshop is organized by the PMEX team. So PMEX is short for Plant Method Exchange Platform. And we developed this interactive workshop series to connect the plant research community with experts which are very good at certain research skills. And here is showing you the organization team, like for this workshop series. So we have Xiaohui from Caltech, Jin Chang from UC Berkeley, Mingjie from Duke, Vincent from UC Riverside, and Catherine from Wisconsin Madison. And for our series, so it started from June this year. It's about the last workshop is about plant transformation. And today we will focus on the MorphoGraphX assisted quantitative analysis of plant morphogenesis. And if you're interested in like post-translation modification or confocal microscope imaging, so we will have 2 upcoming workshops happening early next year. So please mark this on your calendar, and if you're interested, like join us again for the upcoming workshops. And for today's workshop, so we are honored to have Mingyuan and also Shuyao to join us as a panelist. So the overall schedule would be the first talk and a demo presented by Mingyuan for 25 minutes, and then we will do the first Q&A. And then Shuyao will do the second talk in the demo, and we will move to the second Q&A. And if you have any questions about the MorphoGraphX or their research, feel free to post your questions in the Q&A like at any time, and I will ask the question to the panelist. And we will also provide attendance certificate to you, and together with a recording link. So I will let Mingyuan to share the screen.
Okay.
Mm-hmm.
All right.
Yeah, so Mingyuan is currently an assistant professor from UIUC. She did her PhD at Harvard with Dr. Elena Kramer, and then she went to UConn did her postdoc with Dr. Yao Yuan, and he only did like 3 years postdoc there. And he had a very beautiful recent work about the symmetry breaks during the early flower development. And today she are going to present us about how MorphoGraphX can help us to understand more about morphogenesis in mammalous flowers. Mia, go ahead.
Okay, thank you. So before we talk about MorphoGraphX, let's talk about flowers first, because everyone likes flowers. They're pretty. Their morphological diversity is seemingly infinite. But among this diversity, most flowers can actually be grouped into 2 major types. They can either be radially symmetric or bilaterally symmetric. So the former has multiple planes of symmetry, while the latter only has one, dividing the flowers into 2 mirroring halves. From an evolutionary perspective, the ancestors of all flowers and of all the major flowering plant groups are predicted to produce radially symmetric flowers, so bilateral symmetry is a derived state. The current estimation is that as many as 200 transitions happened between these 2 forms, so floral symmetry is a very evolutionarily labile trait. The transition from radial to bilateral was also considered as key innovations in many groups, especially some of the species-rich groups like orchids, the legumes, because people think that this conformational change in flora form enabled very specific pollinator interactions. So this is also an ecologically very important trait. From a developmental perspective, bilateral symmetry is also very interesting because it can be expressed and manifested in many different ways. For example, it can be changes in organ number, size, shape, color, or any possible combinations between them. But no matter how the flower looks like in the end, the underlying developmental principle in making one is actually the same. Because having one plane of symmetry like this, it means that the left part and the right part of the flower is the same, right? But it also means the upper part and the lower part of the flower followed different developmental trajectories. In other words, regardless of how different these developmental programs are, regardless at which developmental stage the differences are elaborated, or regardless of how the flowers look like in the end, this division between the upper and the lower is a prerequisite of making any bilaterally symmetric flower. It's a universal rule. And this division can be readily observed since the very beginning as well. So flowers are produced by flora meristems, and flora meristems are produced by inflorescence meristem. As soon as a flora meristem is produced, by default, half of its cells will be located relatively closer to the inflorescence, which we call the dorsal domain. And then the remaining half will be located relatively further away, which we call the ventral domain. So this dorsal ventral positional information is the inherent physical property of cells in the flora meristem, using the inflorescence as a reference point. In some species, those cells the cells in the flora meristem do not respond according to their positional information. So organs initiate in the dorsal versus ventral follow very similar developmental trajectories, resulting in radially symmetric flowers. But in some other species, the cells react according to their relative position. So the organs initiate in the dorsal versus ventral follow different developmental trajectories. Therefore, symmetry breaking occurs, resulting in bilaterally symmetric flower. Now that we know the dorsal and the ventral can be readily physically distinguished, it's normal to wonder how are the domains defined. Luckily, work from the early 1990s in Snapdragon have provided a solid foundation for us. So people discovered that a TCP transcription factor, cycloidea, together with its close homologs, they control the dorsal organ identity. So both of those genes have dorsal specific expression, and they control the developmental process of the dorsal organs. Without the cycloidea genes, the flowers become radialized, completely ventralized. So this discovery of cycloidea started a whole intense period of studying of cycloidea homologs in many, many different plant taxa. What blew people's mind was that for every taxa examined to date, there seems to be a correlation between bilateral symmetry and the differential expression of cycloidea along the dorsal and ventral axis. So this cycloidea story also became a textbook example that how developmental novelty can evolve repeatedly across evolutionary scales through the redeployment of the same molecular program. Whenever we hear cool stories like this, it's hard to not ask how. How did such a cool thing happen? Unfortunately, we don't have any answers to these questions because we actually don't even know how is a dorsal specific expression of cycloidea first established in any species. So currently, we have only two potential regulators of cycloidea genes. The first one is auxin. If you know auxin, your reaction is probably, duh, of course it's auxin because it's literally involved in everything, right? But I urge you to hold your beer. So as early as 1960s, people have been applying auxin or auxin transport inhibitor to a bunch of species like those ones, and observed that exogenous application of hormones can lead to symmetry change. And the other potential regulator is a gene called blade on petiole, BOP. So BOP mutants has been observed analyzed in a bunch of species as well, as I show you here. They all have a whole range of developmental defects, but among those defects, one of the common ones being changes in flora symmetry. So either the auxin application or the BOP mutants, the phenotypes clearly indicates that those two factors are involved in the regulation of cycloidea genes. But how are those involvement implemented at a molecular level is not known, because we're all facing the common bottleneck that phenotypic effects can be really hard to translate into molecular mechanisms. And to understand the molecular mechanisms, we need systems that have bilaterally symmetric flowers, but also equipped with proper tools. And this moment did not come until recently, and here's my very cute mimulus flower. A bilaterally symmetric flower, but also can be easily transformed, so a truly emergent model system. And now we can use Minimalist to really address those mechanistic questions regarding symmetry breaking in making flowers. So we have two paralogs of cycloidea in Minimalist, 2A and 2B. Both of them control dorsal identity redundantly. If we overexpress either of them, the flowers become kind of dorsalized. And if we knock both of them down, the flower becomes completely ventralized. To understand everything more closely, we put a few things in the plant. So firstly, plasma membrane marker to see the cells and the tissue shapes more clearly. And DR5 as a synthetic auxin reporter that report as the site of high auxin, as well as native reporters of Cyc2A and Cyc2B, translational reporters. As a proof of concept, so here we have two florobuds that just finished initiating all the organs. So all the bumps are the organ primordia. If we look at DR5, you can see that it's quite similar to what's known in Arabidopsis already. It's concentrated on the tips of the primordia or as well as lateral organ margins. But for both of the cycloidea genes, their expression is restricted only to the dorsal part. So now what we want to do is to go back or earlier in time to see when and where are those two genes expressed and their relationship with DR5 patterns. To orient you for what we're going to look at, so here is a side view of an inflorescence of a mimulus. And we have an inflorescence meristem on top with a baby flora meristem below it with its dorsal and ventral domain. So the things on the side are from older flora meristems from the previous node, but our focus will be on this baby down here. While searching for the first appearance of cycloidea genes, the first surprise that we ran into was that, unlike what's known in Arabidopsis, the physical initiation of flora meristem in Mimulus is not accompanied by auxin maxima. For example, here we have two different samples, and you can see that the flora meristem is already very physically defined, very round and chubby. And the DR5 signal can be observed in many other parts of the plant, but there's nothing inside of the flora meristem, right? However, at this stage, both of the cycloidea genes already established their dorsal specific expression. So what does this mean? It means that although we tend to think auxin is literally involved in everything, but perhaps not responsible for initiating symmetry breaking in the flora meristem and activating the cycloidea genes. So now we have to go even earlier to try to see how are these two genes initiated or activated. Now let's look at 2A. It turns out for 2A, we have to go way earlier to a stage pretty much the youngest flora meristem that I can physically dissect, which is pretty much look like this. So from the morphology of the tissue, it's hard to even figure out what is what. But if we process the surface curvature of this tissue, you can see that there's a small group of cells down there that's kind of red. That means that they have positive curvature. They're slightly bulging out. And that's the location of the incipient baby flora meristem. This is a very, very, very early stage. And at such an early stage, this is actually the first time that we can observe the expression of Cyc2A. And if we quantify the cells with the highest expressions and outline them in the tissue, and it turned out it's almost all the cells in the flora meristem, as well as some cells adjacent to it, and some cells in the inflorescence meristem as well. And only when developmental proceeds a little more, its expression domain becomes dorsalized. Okay, this is 2A. How about 2B? And it turned out the first stage that we captured the expression of 2B was later, something like this. Like you can see the boundary between the inflorescence meristem and the flora meristem become very visible already. There are several layers of cells that's blue, meaning that they are having negative curvature, so they are concaving in, marking this very physical boundary. Also, unlike 2A, which was first expressed in almost all the cells, the initial expression of 2B was always only at just a few cells by the boundary. And only as the development proceeds, more cells will start to express 2B, and then it becomes dorsalized. Okay, so far we have established that auxin is probably not involved in initiating these two genes, and these two genes have a slightly different interesting initiation patterns. How about BOP? To address that, I constructed some BOP CRISPR lines, and which makes flowers that kind of look like this. So it's kind of dorsalized. And indeed, this phenotype is due to an ectopic expression of both sex genes. For instance, here we have two flora meristems that's still in the process of organogenesis, initiating organs. And it's obvious that both of these two genes are no longer restricted in the dorsal side anymore, particularly 2A that extended all the way down to the ventral side. So now we want to see whether the patterns of activation, initiation of the Sec2A and 2B changed in BOP CRISPR line. Well, the short answer is nope. So the initiation of 2A is as early in the same pattern as well as 2B in just starting out just a few cells by the boundary region at comparable stages. In other words, BOP is also not responsible for initiating symmetry breaking and activating the cycloidea in the dorsal part. Now you can ask me, well, then what's going on? Because people obviously observed those phenotypes, right? So what we think is going on is that both of these potential node regulators are responsible of maintaining the cycloidea gene expression during the process of flora meristem expansion. So just now I told you that DR5 is not really associated with the initiation of flora meristem, but it does show up in the early stages of flora meristem expansion. And once it shows up, it's highly dynamic. So initially it will be a few dots on the central division line, and then it will increase in the center, move to the dorsal, then show up at the ventral, and then the number of cells expressed in DR5 will drastically increase both the dorsal and the ventral, and then resolving 5 points, and followed by physical initiation of sepals. So all these dynamic happens when there's no organ initiation going on, and it's very different from what's known in Arabidopsis. During these stages, I know there's a lot of information to look at, but I want you to pay attention to 3 things. Firstly, during this entire stages, the flora meristem stayed round and chubby, right? There's no organ initiation. Secondly, both of the cycloidea genes remained their dorsal expression domain very consistently, right? And lastly, the expression boundary of DR5 and cycloidea genes almost always perfectly overlap throughout all these stages, as if there's an auxin-dependent pathway maintaining or restricting the expression domain of cycloidea genes, which is very curious. Now let's look at what's going on in BOP. So just now we mentioned that the patterns of initiation didn't change, right? But the overall auxin level decreased a lot in BOP CRISPR line, and you can see the DR5 patterns are quite different from the wild type as well. But it just happens so perfectly that the size, the stage of the tissue when you first start to express DR5, as well as the stage that organogenesis is onset, aligned perfectly between those two backgrounds. And it just happens so perfectly that both of the cycloidea genes start to ectopically express during the stage that DR5 showed up. So we think what we're seeing here is very distinct phases of cycloidea expression regulation. Both of the previous known regulators are involved more in the maintenance and perhaps the specification of these genes, but not in the initiation. So obviously I can't really tell you who is responsible for the initiation of cycloidea expression yet, but we have some good leads, so hopefully you will stay tuned. And as Mingyuan mentioned just now, this paper you can find on bioRxiv, and it has been accepted by Current Biology, so hopefully it will come out soon. But before, now I finished my spiel, let's talk about MorphoGraphX. So I first get to know this software a few years ago during grad school, and it really became a very important part of many of my imaging work. Because you can see, as I demonstrated just now, it's a very powerful tool that helps us to see the information, the patterns, and the numbers that our eyes won't be able to notice. A clear example is this slide, Sure, the first two columns are the maximum projection of the confocal images. It's informative, but not nearly as informative as what we can quantify from the curvature, from the signal intensity. So today I'm going to show you a very quick and short example of how to reproduce one of those panels. And we start with Z-stacks. So for my confocal, we collected 3 channels, the blue, the red, and the green. And here I separated the blue one and the green one, and you just drag the plasma membrane line directly inside. You can see this beautiful 3D constructed tissue already. So after that, our first step is to extract the surface, the shape of the tissue, which we start with by blurring them multiple times. So these are the parameters that you can choose and optimize for your own samples. After we blur them a few times, we do edge detect. Again, this is a threshold that you have to optimize for your own tissue as well. After we detect the edges, we can see we'll do a process called marching cubes surface. So this is pretty much when the surface are actually extracted. And you can see it's like this. So there's a way to show that at this point, we have a lot of rough triangles covering the surface. And here what I'm doing is to remove the part that actually doesn't provide us information. So it's remove so that this can decrease the computation time later in the process. So I train off the bottom, and I do the same exercise for some other tissues as well on the side. And here, we train off most of the things and we save the mesh. So that's one of the most important things while using MorphoGraphX is that there's no undo button. So for each sample, I will have multiple versions of meshes saved for different steps. Now what we're doing is to smooth and subdivide the meshes multiple rounds so that we can get the resolution that we want. So you can see I have been repeating this process. And after the last subdivide, I changed the smooth path from 1 to 10 to get a smoother surface. And now the processing of the surface is done. Save again. And save it in an informative way so you know which mesh is responsible for which step. And then we do, oh, I think I skipped. Let me go back one slide. Okay. Now we can project the signal from our actual Z-stack onto this mesh that we processed through a process called project signal. So this is a process you have to experiment different numbers for your particular sample. I'm showing here that different numbers have a very different effect for the projected signal, right? So just now 4 and 6 is too distorted, 1 to 2 is very light. So the general rule of thumb is that I keep it consistent for all my samples across experiment. And after we project the signal, now it's a fun part, which is, where's my mouse? We segment the cells. So auto segmentation normally for samples like this works really well, and there are only a few things to change. So after I change them and we run, you can see how it's automatically processing blurred and seed of the cells and then segment. And lastly, it will correct a few errors. So this is one of the examples after auto segmentation. You can see some part, and save. Some part of the cells is segmented really well, but some other parts are not. So after we save this mesh, we will have to correct the errors. And I will show you a few, correcting a few errors is actually a really fast process for a small sample like this. So we can compare the mesh and our original sample, and then start to select the cells that we want to correct. You don't have to select too many things inside, and then now we can just extend to the whole cell and clear the segment. So now if we bring back the surface, you can see that those cells do not have the labels, the colors anymore. Then we manually seed those cells by just drawing some outlines inside of the cells and rerun the process segmentation. Now the errors are corrected. So then you save it again. So we do this exercise a few times to correct all the visible errors, and we end up with a sample like this. So the last step is that we're gonna remove the edges, because you can see the edges are really messy and it doesn't really have cells to be segmented. So we want to clean up everything before we do any analysis. And this process is quite similar to what I just said. You select some things, you extend to whole cells, but instead of clearing the segmentation, I would just delete it using the scissors on the side tool. And in the end, we end up with a sample like this. And here's where the fun part begins. So now we can do analysis of the cells. Oh, and save, of course. Save it informatively. So for example, with a sample like this, we can do something. We can do a different kinds of heat map. Let's do a basic one, such as the cell area. So you can see instantly come out. For different kind of heat maps, you can adjust the scales of the heat map. So normally for any kind of heat map for one experiment, I will keep the scales consistent. And then we can all do different kind of aesthetic things, like change the border color of the cells, now it's black, or the style of the heat map, for example. Okay, and then I want to show you how to project the M turquoise signal onto this processed mesh. So just now you see that I have two different separate Z-stack channel. What we're doing here is gonna, oh, sorry, let me skip this. So now we're just dragging the M turquoise stack directly onto this mesh, and nothing is showing up right now, right? Because just now we changed the border from blue to black, and now we can change it back. And now with the M turquoise stack, we do the same exercise of projecting the signal onto this processed mesh. And for now, for this second projection, the numbers can be adjusted according to your own samples. After that, we measure the signal intensity by going to heat map, and there's a function called signal interior, and voila, that's our heat map for the signals. And then we can do some aesthetic things again, or change the borders. Yep. But the one last thing I want to show you is how to highlight the cells with the highest expressions. So if you look at upper right corner, we can actually save every information in this heat map into a CSV file, which is what's happening right now. And this CSV file will include the signals for each cells. And after that, if we open this file, okay, if we open this file, you can see there are three columns. The first one is the label. That's just the label identifier for all the processed cells. In our sample, we have 314 cells. And then the third column is a relative signal intensity. Since we want to highlight the cells that with the highest expressions, so here I'm just try to calculate what is the 90th percentile of signal among all these processed cells, which is a number of 2,866. And then we go back to this heat map to use a function called heat map select. For the lower bound, we just calculated, and the upper bound just put in some ridiculously large sample, and voila, the high cells with the highest expression is selected here. So now we can do more aesthetic things. And the last part to recreate that figure is that we want to rerun the curvature calculation again, which we go back here and go to the curvature part, compute the tissue curvature, and that's the radius is something that you want to adjust for your own sample as well. And there are different types of curvature, and I have been using Gaussian surface curvature consistently. And for all the curvature maps in the study, they have the same heat map scale as well. So now I'm setting that scale, and then finally we change the appearance of the heat map. And done. Okay, so I know I ran through everything really, really quick. So before I answer any questions, I want to thank Daniels, Mingyuan, Richard, Soren for teaching me. And MorphoGraphX has a very active forum online as well. And thank you very much for your attention.
Great. Yeah, thank you, Mia, for showing like the workflow very beautifully. And we do have one question from the audience. It's like, so the audience is very interested in knowing how to optimize the confocal image quality for the better service reconstruction in the MorphoGraphX. So do you have any suggestion when you collect the confocal image for that?
I have a few from personal experience. I would just say it like this. Firstly, it depends on whether you're staining your samples or you have transgenic markers. For example, sometimes we stain samples with PI, it almost never work as well as plasma membrane lines. So the staining or the signal itself is something. And then for collecting the confocal image, it's a trade-off between quality and time. So sometimes if your Z-stack is very, very fine, the quality of image will by default much better. But again, if you're doing live imaging, you probably don't want to spend that much time on the confocal for your laser to zap the tissue. So it's a delicate balance. And I think it will, for each sample is different. So you have to kind of experiment different types and length of time to figure out.
Yeah, thank you for the answer. I think we need to move on. So and thank you again, yeah, for the great presentation. And so Shuyao, I think you can start sharing your screen.
Okay.
And it's a great pleasure to introduce our second panelist. So Shuyao Kong, he did his PhD in plant biology in the lab of Adrian Yoder. He published multiple first author papers in Rotor Lab on Arabidopsis flower development, and I think that should be the major thing he want to present today. And he's now a postdoc at Harvard Medical School under the supervision of Dr. Olivier Pauquier. And so, Shuyao, the stage is yours.
All right. Thank you so much, Mingyuan, for the introduction. So today, I apologize in advance. I will not be able to show how to process the images using MorphoGraphX because the lab computer broke down. So I will just share with you how I process the signal in my newest paper. So the paper is now online in Nature Communications. So stochastic gene expression in auxin signaling in the floral meristem of Arabidopsis thaliana. So I will walk you through what we found and how we use MorphoGraphX in this publication. As you know, auxin signal is very important for plant development. And so I also study sepals. So I studied sepals. So sepals are these structures that grow from the floral meristem. As you can see, this is a picture of the Arabidopsis inflorescence meristem from which the floral meristems develop. And as also you can see from this picture, here I have an auxin signal marker, which is DR5, driving the expression of M scarlet 1, which with a nucleotide. And you can see from here, this is the stage just prior to sepal initiation from the floral meristem. And auxin signaling is highly concentrated in the 4 places which are going to give rise to the 4 sepals, and that's also shown in the schematics here. So although auxin signal is very important and focuses on these auxin maxima just prior to sepal initiation, we found it actually has a lot of noise. So this was the stage that I just showed you in the previous slide. If you go back from this stage, this is a live imaging. you can see in this early stage, auxin signal is actually very noisy. It sometimes forms these randomly positioned stochastically positioned auxin noise with just a few cells with very high DR5 signal surrounded by cells with lower DR5 signal. But this noise is eventually going to canalize to these 4 places with very high auxin signaling just prior to SIPO initiation. So that was very magical that we found, and we wanted to figure out how it happened. So in order to visualize auxin noise, we adapted this dual reporter system where in the same cell, we introduce 2 markers. So both of them are driven by the DR5 promoter, and they're driving the expression of two fluorophores with different colors. So one of them is yellow, basically Venus with a nuclear tag, and the other is M scarlet I, also with a nuclear tag. So the idea is if you look at this image, if there's absolutely no molecular noise in the expression of auxin responsive genes, in all the cells of an auxin maxima, there should be equal amount of Venus signal and M scarlet signal. But that's actually not the case. So we found that in some cells, there are more scarlet signal. In other cells, there are more Venus signal. So the expression level of the 2 markers, even if they're driven by the same promoter, is actually not equal. And that is what we call stochastic gene expression. So how do you quantify this thing? So this is showing the result of the quantification. So basically for example, at stage 2B, there are 4 auxin maximum, but what we actually found, so when you look at the pattern at the whole flow meristem, the pattern is actually quite robust and precise. So auxin signal highly concentrated in the 4 incipient sepal primordia. But when you zoom in on one part of it, for example, when you zoom in on this part, which is the incipient inner sepal primordia, when you look at the Venus signal versus the M scarlet signal, actually the signals are quite different. So for example, there are these 4 cells with absolutely no expression of M scarlet, but they actually have strong expression of the Venus reporter. So we know from this quantification that auxin noise is always there from the beginning to the end, but eventually somehow the meristem can produce these 4 auxin maxima despite the molecular noise. So that was very, very interesting. And now I will show you how this is quantified in MorphoGraphX, how to quantify the auxin signal MorphoGraphX. So basically, as I told you before, in these plants, we have a dual reporter. We have DR5 driving Venus. We have DR5 driving M scarlet, and both of them are nuclear localized. In order to extract the position of the nuclei, we also had a nuclear marker, basically H2B TFP, which was driven by the P18MAL1 promoter, and that's epithelial. Yeah, that's epithelial promoter. So from that, from the H2B signal, the first step that we did was to create this L1 mesh. So you can see here each nuclei is labeled by a little ball, which is a mesh in the MorphoGraphX. And so how we did that, we imported the H2B TFP signal into MorphoGraphX, and then we blurred it. We did a Gaussian blur. And then we did a extract local maxima command, which gave us the position of the center of each nuclei. And then from these centers, we created basically like a ball mesh around each center. So that's what you're seeing here. So from the L1 mesh, we know the position of each nucleus, and we have the DR5 Venus stack, and we have the DR5 Scarlet stack, and so we are able to project each of these signal into the L1 mesh, and that is going to give you a heat map of the Venus signal and the heat map of the M Scarlet signal. And so also as Mia previously showed, you are able to export these heat maps as a CSV file. And so the CSV file basically contains the positional information where each nucleus is, and also how much Venus signal it has, how much M Scarlet signal it has. So from the CSV file, we could do downstream processing, for example, calculating the agreements versus the divergence between the Venus and the M Scarlet channels and so on, and calculate the molecular noise. So basically, that was a brief walkthrough of how this signal was quantified. I apologize again for not being able to show it on in real time on MorphoGraphX because of the computer issue. But basically, I'll tell you what we found. So returning to the image that I showed in the beginning, where there is persistent stochastic gene expression coming from an auxin promoter, auxin responsive promoter, and there is a lot of auxin noise at the beginning. But just prior to sepal initiation from the meristem, somehow the auxin signal canalizes into these four places. How the plant does it, so it has something to do with cell number. Because in the beginning, the meristem is relatively small. Whatever cellular noise that is present in the beginning is not tolerated. So basically, it shows up in the global pattern. But when the meristem grows bigger and more cells are added, and so in stage 2B, which is just prior to sepal initiation, the stochastic noise in cell neighborhoods are actually averaged. So that basically means if a cell just happens to express a lot of DR5 and its neighboring cell is not expressing it at all, it doesn't matter, because when you look at the whole cell neighborhood, there are always going to be some cells that express the auxin response, and that's going to make the region into an auxin maximum. So that was basically what we found. And so if you're interested, you're welcome to check our paper on Nature Communications. Thank you.
Okay, that's short and sweet. And Shuyao, thank you for the good presentation of your recent work. And we do have some remaining questions from the registration link. And for the for the audience, if you have any questions, feel free to ask to put a question in the Q&A. Yeah, we have one. Yes. So here is here's a question. So for cells that are deeper below the surface, how can you view them so it's easier to correct their segmentation?
Yes, so for the purpose of this paper, we only looked at did I say epithelial? We only looked at the epidermal cells. So like the cell on the L1, but if you want to look at the deeper layer, like the L2 and L3, what you can do is you can use like a ubiquitous nuclear marker. So in our paper, we use the ATML1 promoter to drive the H2B, but you can actually use a ubiquitin 10 promoter or the 35S promoter. And once you get that, you basically get nuclear signal in L1, L2, L3. To specifically look at L2 and L3, not L1, what you can do is you can create a surface from the H2B signal, and you can annihilate from that surface. So basically you can eliminate the signal that's within a certain depth from that surface. So in the case of flow meristem, this is roughly 6 to 7 microns. So once you eliminate H2B signal from that surface, the only surface that's remaining is L2 and L3, and then you can use that to segment your cells.
Yeah. So here's another question is like, so like the audience is curious about like, so for the same genotype, sometimes it could have like a large variation. For example, you're also showing like, the variation of like the gene expression, like across different cells. So do you think like, how do you think like the morphographics assisted quantification can help to analyze and also to better understand this kind of variation across different like replicates and different cells?
Yeah, so if I understand correctly, the question is about how can morphographics quantify, for example, DR5 or auxin signal and compare between different genotypes.
And also like sometimes or even for the same like a genotype, it could have a lot kind of a large variation. And how could like morphographics help us to better understand this thing?
Yeah, thank you for the question. That's indeed very insightful. So what you can do is you can, for example, if you think that even within the same plant or within the same inflorescence, there are gonna be differences between samples, you can image all of them and then you can quantify them in morphographics. And so one thing I didn't show, actually, I think I can show quickly. One thing I didn't show in the paper, in the presentation, actually, I think this is a good example, is you can quantify different replicates. See, these replicates are the same genotype and the same treatment, and they look different. I mean, the overall pattern looks the same, but when you look at the cells, like this auxin maxima looks slightly different from this one. What you can do is you can register, because from the morphographics quantification, you export the CSV file, right? And then from the CSV file, you get the coordinates. You can get either the XYZ coordinates of each cell, or you can get the polar coordinates. And from these coordinates, what you can do is you can register them. Basically, you can register them in R. You can normalize by, say, the radius of the bud, and then you can bin the signal into these tiny little bins. So this binning is gonna allow you to compare between different replicates, and you can also calculate. So for example, at this particular position, what is the mean signal? What is the average signal between replicates, and what is the standard deviation of signal between replicates? And you can also, because you register the signal in this coordinate system, you can compare between different conditions and see, for example, here, this mock treated condition is very different from the original treated condition. And that's one major finding that we had.
Okay, yeah, thank you for the answer. And here I think there is kind of a more practical question, I think, for both of our panelists. So some people actually complain and say like it seems hard for us to explore the full function of the morphographics, and sometimes like they cannot find a very useful protocol, or like they cannot find a detailed description of how to do like certain like like analysis they want. So for you have used the morphographics a lot. Do you actually identify any useful resources for that? Or do you know, any way to really help the community to be more familiar with like morphographics and to do whatever they want to do?
So I think for me,
Shuyao, you can start,
I guess the most efficient way is to bring your project to Richard Smith Group and do an internship. Or so in my case, I didn't have a US visa, so I couldn't enter back into the country if I did that. In that case, I contacted him and then consulted my project, and he gave a lot of help.
And Mia, do you have any suggestion?
Yes, because I didn't do an internship with Richard Smith. The forum the link for the MorphoGraphX forum that I put by the end of my presentation, I used that a lot. And both Richard and Soren, they are very active on them, and they pretty much really help you to really solve the questions. So I do use that a lot. And another resources, which is why I agreed to do this workshop, is you need to know who are familiar with this software. For example, in my case, people from Adrian's lab, like Mingyuan Zhu. I found talking to people and look at their publications of what kind of analysis they did would be incredibly helpful to help you to structure what kind of analysis or experimental design that you want, and then start from there. And if you run into trouble, ask people who have done it or used that forum, and most of the time the issue can be solved.
Yeah, I mean, I'm glad, like Mia, you just mentioned this. That's also the point of like this workshop. It's like if you are very interested in like cell size quantification, like nuclei signal density quantification, now you know the experts who contact. So, and here's kind of the last question. Like, I mean, the last question for today's workshop. Some people kind of is kind of curious about like scale up or expand like the use of like the MorphoGraphX. So he's kind of talking about, okay, can we actually apply like the MorphoGraphX analysis to some another existing system, for example, like measuring like algae growth or biofilm, like a moss, or like just different like kinds of like samples. So I want to hear your insights about like, since you have used MorphoGraphX, do you think they have the potential to do such thing? Or like, if so, like, is there any like, idea about how to expand this technique?
Mia, you can go.
I think the answer is yes, and as a fact, I think the developers are actively exploring these areas as well. I know people study algae and also different animal systems in Drosophila, and others have been using this tool. So their goal, although right now relatively small number of researchers have been using MorphoGraphX, but I think the goal for the developers are definitely expand outside of the model systems. And I know they are also actively developing 3D segmentation, which there are some publications out using that already. It's really cool. So the potential is infinite, and it's just a matter of when is it going to come out, and how are we going to learn it and apply it to our own research.
I think I agree. I think that the software does not, I would say, although I didn't work on non-model systems, I would imagine if you have like a cell wall stain, like if you want to study algae and ferns, if you have like a cell wall stain or a cell wall marker or something, and then you just image a 3D stack, and then I think it should be analyzable.
Okay. Yeah, and there's a comment from the audience say like, the audience is very interested in like, some how to connect the MorphoGraphX quantification, for example, with R programming. So like, Shuyao has some experience of like how you can like really use R programming to help you to calculate the sepal again, the petal length. So, feel free to contact the Shuyao for the details. Yeah. And I think that's it for today. Thank you so much for the participation, and especially thanks to Mia and Shuyao for like your nice presentation. And so the link of the recording, as well as the contact information for our panelists, and will be sent to you via emails later. And thank you again for the participation. Okay.